We are going to end this series of notes on chromosomal mutations. And a chromosomal mutation is different than a gene mutation. A gene mutation only affects one DNA letter, maybe two, but you're looking at just a piece of DNA. In a chromosomal mutation, you are actually going to be changing the number of chromosomes that you have. So 46 is your normal. You could have less, you could have more. That would be a chromosomal mutation. Or you're changing the structure, the shape of the chromosome somehow. You're not affecting just one DNA, A, T, G, or C, but in a chromosomal mutation, you'll actually be affecting whole pieces of DNA with these chromosome issues. So we're not looking at the DNA or the gene, but we're looking at the whole chromosome. And we're going to take first a look at just how you could change the number of chromosomes. We know that 46 is the correct amount, 23 from each parent, but there are people who have the wrong amount of chromosomes. They have too many or not enough. And how does that happen? How do you get the wrong number of chromosomes? And the way this works is by non-disjunction, and I'll show a picture here in a second. But during meiosis, your homologous chromosomes are supposed to line up side by side. One gets pulled to the left, one gets pulled to the right. But in non-disjunction, they don't move away from each other. They stay linked, or they stay connected. So here you can see right here the non-disjunction. This is what should happen. One chromosome gets pulled one way, one gets pulled the opposite direction. And that way when the cell divides in half, we were supposed to have 2 chromosomes in this cell, and we were supposed to have 2 chromosomes in this cell, but that's not what ended up happening. We ended up with 1, 2, 3 chromosomes over here on the one to the left, and only 1 chromosome on the cell to the right. So you end up with a gamete, which should only have 2 chromosomes, but this one has too many. Okay, so we're increasing. And these gametes don't have enough. So depending on which gamete is being passed on to the child, you could have a child who's been, shortened a chromosome, or you could have a child, if it inherits one of these first 2, that has an extra chromosome. Now, the easiest way to decide if you have a chromosomal mutation is by a karyotype. Now, this slide may or may not be in your notes. It's something that I've added recently. So if you do not have this in your notes, you may want to jot this information down off to the side. You're still required to know it. Even if your notes aren't fully complete, you need to then add this in. So a karyotype is a picture, and it shows all of the chromosomes that are in a single cell, and it organizes them by size. So here would be an example of a karyotype, and it organizes them by size. So it's going to put the largest ones first, and it's pairing them together. So notice you don't have 46 pairs. You have pair 1, which has 2 in there, pair 2 has 2, and pair 3 has 2, and everything should have 2. Your sex chromosomes actually go at the very end. So you're going to have 22 pairs, and then your 23rd pair, this one right here, notice that you have a very large chromosome with a smaller one. So this is an X, this is a Y chromosome here, an X and a Y, because we have 2 very different sizes. If it was a female and she had 2 X's, these would be about the same size. They would look pretty identical to each other. So you can also tell gender from a karyotype. And you should have 23 pairs of 2 each for 46. Now you could go through and count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You could count all of these chromosomes, or even if you just glance through here, hopefully you see that there is a mistake. This karyotype has a genetic mutation. Pair 21, I shouldn't say pair because it's not only 2, is called trisomy. There are 3 number 21 chromosomes, and whenever you have 3 of chromosome 21, that is how someone has Down syndrome. So if you know anybody with Down syndrome, that is how that happens. It's a genetic disorder caused by having 1 too many chromosomes, and specifically in this 21st spot. So if you have maybe 3 number 13s, then that's not going to be Down syndrome. That would be some other genetic disorder. So it does matter where the mistake is at as to what genetic mutation you have. But this would be an example of having too many chromosomes. Now let's take a look at the structure of your chromosome. So you can have 46 normal chromosomes, but the structure of them is incorrect. So we have a specific one here called deletion. So you can see the chromosome here should be about that size, and all of this information here are different genes of DNA that code for a trait. And then deletion just means a section of that DNA, whatever section it is, whatever section of the chromosome is cut out, pulled out, and so the the halves kind of recombine here, so you have the rest of this DNA that's good and is there. But that chromosome is definitely different in structure than its original because it's lost a good portion of information there. So deletion is when you lose a piece of a chromosome. Now we did have deletion in our other type of mutations as well, our point mutations. So make sure you read your questions carefully, as when it mentions deletion, it will make sure to talk about whether it's a chromosome mutation or if it's a gene mutation, and that's important since deletion is you have an example of deletion in both types. We also have another type of chromosome mutation, and again, we're not looking at the amount of chromosomes but mainly their structure, called duplication. And this is where a piece of your chromosome is repeated. Now they broke these up into like sections. So this is one chromosome here, but they broke it up into section A, section B, and C, just so that we could see how this works. But in duplication, a piece of it is going to get like copied and repeated. So this CD portion right here, we're kind of inserting another CD portion, so you have that genetic information twice on your chromosome instead of only the once that your body needs. It has extra information, so it kind of slows things down trying to read through all this extra. Another type of chromosomal mutation affecting the structure is inversion. And with inversion, a piece of your chromosome just gets reversed. So here's the normal order, A, B, C, D, E, F, G, H, I, and a piece in the middle here is just getting twisted. So this section right here, not in alphabetical order anymore. So as your body is trying to read this DNA, because this is a chromosome, there's DNA inside of this chromosome, and now all the letters are going backwards. All the letters are in the reverse order. So the stop codon and the start codon, all those things are backwards now. So that can cause quite a few issues. And then the fourth type of chromosomal mutation is called translocation. And that just means that a piece of a chromosome breaks off and attaches to another one. So in this one they color-coded them so that you could see that a little easier. And they also have the letters on here. And you can see that the red and the blue have switched places. I want to make sure that you understand that this is not crossing over. Crossing over is something that is supposed to happen, and it happens between two homologous chromosomes, one from mom and one from dad, that are similar. This does not. Okay, this type of mutation, this translocation, first of all, is not going to be between homologous chromosomes, so it's between non-homologous. Yikes. Non-homologous chromosomes. So if we go back to that karyotype picture, it might be taking one chromosome from pair number 1 and one chromosome from pair number 8, and they switch places. That should not happen. The two chromosomes that are in pair number 1, those are homologous, and it's okay for them to switch pieces. That's crossing over. But in translocation, that's not crossing over because the chromosomes are not similar, and you don't want them to be trading information unless they are homologous chromosomes. Alright, so to finish us off, some significance of this. First of all, many mutations have little effect or even no effect on the gene being expressed. So the phenotype of the individual is not always going to be changed. Mutations that are in your somatic cells, like your skin cells, brain cells, so on, those cannot get passed on to future generations, only if the mutation is in a gamete. So I guarantee that that is something that you need to know. So people who do a lot of sun tanning and they maybe get melanoma, skin cancer. If that skin cancer is from the extra exposure to the sun or to, the tanning bed, whatever it might be, then that's not part of their gamete. That's not something that they can pass on to their children, and their children have a higher chance of getting skin cancer. If the skin cancer, is in the DNA itself, in the gamete of that mom or dad, then they can get passed on. But somatic mutations that happen during your lifetime are not going to get passed on to your children. Although some mutations can, cause a lot of harm with the sickle cell, Down syndrome, things like that, they're also a source of genetic variety. So yet another way that your DNA is not the same as someone else's. And that wraps up our information about mutations and transcription and translation.